I've been working all of my scientific life on the question of how the brain gets wired up properly in development. How is it that neurons make connections with the right neurons?
Prior to our discovery, it was thought that all the proteins were made in the central part of the neuron, the cell body, and shipped out, a central factory, sending out trucks.
But what Ozzy, Christine, Kelsey, and I showed is that the machines and the messenger RNAs, the templates for proteins, are actually localized explicitly out in the dendrites and axons, where when synapses are activated, proteins can be made on demand and can give rise to changes at synapses that store information, like memory.
So each neuron has a single cell body and a single nucleus, but can form thousands of synapses. By being able to change the structure and function of individual synapses, that massively enhances information processing in the brain. And understanding how that happens through the local translation of RNAs at the synapse, enables us to have a deeper understanding of how the brain functions.
And our hope in the future is that these basic findings will help with therapies.
And perhaps how synapses can actually be regrown after injury.